We have just a couple of slides, today in our deck, just to kind of help the discussion. Then our speakers will be sharing their own screens, but we'll definitely make sure we share slides after the meeting. You've already received the link to the agenda, but we'll make sure you have that as well. This is our third phenotype across the Innovation across the Phenotypic Translational Divide webinar. This is our third one. I should have it done by now. And this one is focused on cross-species, genotype, phenotype analysis, things we can do to really facilitate those analyses between the very important model organism work and bringing that back into the clinic and how to inform, human-based research. We talked about model organisms in several of the meetings we've already had, but we really wanted to dig into that topic for today. I think more than just who is on the slide was involved in trying to organize this meeting. So just thank you to everyone for bearing with us as we try to assemble a very diverse and somewhat complicated agenda. And again, of course, we'll hear from some key presentations. We'll do a couple panels and we'll do a brainstorming session at the end. And with that, I will turn it over to Melissa Handel to give our introductory talk.
Terrific. Thank you so much. Okay, let's see if I can get my screen to share. Here we go. And move all you people away. Here we go. Okay, can you see my screen?
It's loading.
I lost it.
There we go, yeah.
Okay, terrific. So I'm really excited to introduce our discussion today, and thank you all for attending. I think those of you who know me know I have a great love of all organisms, and I fundamentally believe that there is something to be learned about the relationships between genotype, phenotype, and the environment, and the life history of each organism to inform human health. And we, in the Monarch Initiative and in the Kids First program, have really sort of taken this to heart and are really trying to work hard on thinking about the best ways to do that and to really leverage the wealth of expertise in the organismal community, which includes not just model organisms, but even atypical organisms used in research. So let's see. So I wanted to talk a little bit about some of those types of organisms that are, maybe not seen as, common model organisms, because they really do provide these key insights. And so, we really want to, use the right organisms to understand human disease based upon the phenotypic features and the underlying molecular pathways. And so these are just some really fun examples. For example, the dog's retina has an area centralis, which is analogous to the human macula, and a fovea-like region, which is similar to humans and is useful to study naturally occurring cone diseases. Aged cats are natural models of Alzheimer's disease. They have the same sort of neurofibrillary tangles and neuronal loss. Really interesting, as maybe commonly in the news, naked mole rats don't get cancer. Why is that? Armadillos are a natural host for the mycobacterium that causes leprosy and is the only organism besides humans. Tree shrews, glioblastomas are morphologically and genetically similar to humans and more similar than mouse models. Great pond snails are great models for inflammation-mediated memory dysfunction. And silkworms are a model for uric acid metabolism. And so, this is just, a smattering, and there just are no end to the particulars that allow us to understand that fundamental relationship between genotype, phenotype, environment, and the life history of organisms. With the advent of CRISPR technologies, we have the potential to potentially leverage these types of organisms to, examine these types of genotype, phenotype relationships. But even in naturally occurring lines that, we can still learn a lot mechanistically from these organisms. And so just taking a more holistic, pan-phylogeny view over a fundamental understanding of the relationships between genotype and phenotype. So So how do we do this? So, the central dogma is that, genes plus the environment equals, various phenotypic characteristics or traits. But it's obviously a lot more complicated than that. There's many, many different kinds of interactions that are not just causal, but all kinds of different modifiers. It's actually somewhat iterative and changed over time. There's community population level interactions. And so no shortage of different types of interactions. And so the problem, though, of course, is that the standards for encoding and exchanging not just the genes, environment, and traits, which are challenging enough, and many of us in this group work on those things, but the relationships between them. How do we actually build computational models of the relationships between genes, environment, and traits, such that we can inform how we use those models in the context of diagnosing or otherwise revealing mechanisms of human diseases? So model organisms really matter to patients in particular. This data is a little bit out of date, but essentially over there on the left, we have, there are, at the time we did this analysis, 19,218 human coding genes, and roughly 3,932 of them, or 18%, had phenotypic characteristics that were causal mutations related to those. If you take the orthologs of those 19,000 genes and you look at the 5 most commonly used model organisms, worm, fruit fly, zebrafish, mouse, and yeast, and we look at the phenotypic consequences of mutations in those genes and combine those with what we understand from humans, we end up with an 82% coverage of the human encoding genome. So there's an enormous amount of phenotypic information available about genes in model organisms that we don't know as much about in humans. And so it's really important that we come up with ways to leverage these data in a computable way, as well as in just an easily accessible way. It's not efficient to search, sift through the literature for each one every time you have a patient in front of you. And so income ontologies, and as if you know the Monarch Initiative team, you know that we develop a lot of different kinds of ontologies. And so that for those who are less familiar, this is just a quick overview. An ontology is a knowledge classification that is comprised of terms that are logically defined and logically related to one another, such that computers can reason over them. They really allow the ability to compute over potentially seemingly different types of information and knowledge based upon those connections. And again, that comes back to those connections between genotype, phenotype, and environment that I showed on the earlier slide. And so, we work on a number of different ontologies that are all sort of interoperable and work together, such as the Uberon anatomy ontology, the gene ontology, the NCBI taxonomy of species. And here's just some examples of how some of those kinds of things are related. And so under the hood of a lot of the terminologies that you might be more familiar with in the clinical setting, such as the human phenotype ontology, underlying all of those are logic that relates basic anatomy and gene function and things like that. So this is what that kind of looks like from a cross-species perspective. And we have basically 2 strategies to harmonize anatomy and phenotypes across species called Uberon and UPheno. And we really love the umlaut in our group. So, and whenever possible, we make an Uber thing and we get to use another umlaut. So we have 2 examples here. The one on the right is shown is the Uberon graph, which relates these anatomical structures, the alveoli of the lung, cellular parts, as well as tissue and organ parts, relating them to development, as well as to homology across species, here to fishes at the top left, as well as to gene function, such as respiratory gaseous exchange. And so in this context, we can actually have a computable temporal representation, multi-scale spatial representation that can be sliced and diced for different taxons, different domains, such as the brain or the cardiac portions, or for specific applications, such as in a data collection form or in a user interface. And so these 2 resources we'll be hearing more about later. Just really thinking about them as assets to be configured for your own cross-species needs. A lot of community members from many different organismal communities worked very hard together for many years to build these resources. And it's really a great community partnership and testament to the across, the sort of cross-species community to build this kind of interoperability together. And so with that, I will hand it back to you, Valerie and Nicole.
Great, thank you, Melissa. That was a great introduction. Okay, so next we're going to move it over to the use cases on how structural birth defects and childhood cancer researchers use model systems to inform genetic discovery. And first we'll have Matt Wheeler speak on cardiovascular diseases. Perfect, thanks.
All right, great. Well, thanks, Melissa, for the great introduction, and thanks everybody for putting up with a cardiologist, an adult cardiologist, no less, talking to you on a Wednesday morning. And apologies for any bombing sounds you hear behind me here. It's just some avalanche control going on. When we think about cardiovascular phenotypes, I tend to think of the heart as necessary, but our Drosophila fans know that that's in fact optional. And so this is the heartless mutant, and there's several other Drosophila mutants, including Tin Man and other Drosophila variants that don't have a heart tube, and that's the tube seen in panel A that's sort of missing in F and G. And so Drosophila has been used for many years to look at the cardiovascular system. Of course, Drosophila only has a heart tube. It doesn't have many of the structures that we know and love as cardiologists. But the phenotyping of Drosophila hearts has come a long way, and these are some images from my Baymate when I was doing my PhD. He was doing his postdoc making mutants in Drosophila and showed that you could phenotype these animals just like you can the contractility of the human heart using echocardiography. You could also do survival analysis and exercise testing, and so that middle graph is actually basically a Drosophila exercise test, and then on the right is another group looking at a heat-inducible mutation in a sarcoplasmic reticulum channel gene, showing that you could also see arrhythmic phenotypes. And so many investigators are now using Drosophila to model single variants and can also look across the vast spectrum of existing fly models for evidence of cardiac defects. Importantly, unlike your human survival curves that may take 20 or 60 years to generate, this is you can generate survival curves over 50 or 60 days after birth of your flies. Recently, in the last few years, we and others have been a part of the Undiagnosed Diseases Network, which has a model organism screening core as an important component. We were involved in a case where we had two unrelated patients who had a periodic severe lactic acidemia. Our patient had cardiomyopathy. The other group's patient had transient stroke-like episodes, which was suggestive of mitochondrial disorder. They had extensive genomic testing that was unrevealing, and each was found to have a homozygous missense variant in the gene ATP5F1D that had not previously been associated with human disease. And we sort of modeled the predicted protein structure as shown here, and then asked our colleagues at the model organism screening core if they could model these variants. And so they looked in the Drosophila genome, and indeed these variants were conserved through Drosophila, and the gene ATP-SIMD was the Drosophila ortholog. Then they basically expressed RNAi on different chromosomes and found that they could not generate any phenotypal animals. And so they went to a conditional regional loss of function approach. And so using a head-restricted promoter basically showed that loss of this SYN delta did in fact significantly impact head development, as you can see here in panel C, but we could rescue this RNAi with the human version of this gene. And so the eyes and antenna still had defects, and they had defects similar to other partial loss of function genes that were known to be associated with mitochondrial disease. And so the model and screening core can use Drosophila models and also has C. elegans and zebrafish techniques to try to recapitulate single variants or even a combination of variants in a few cases direct from patients to model human disease. Dynavirio or zebrafish has been used for many, many years to explore cardiovascular developmental genetics. Although zebrafish only has 2 chambers, they have all the critical components of the cardiovascular system, and the development mirrors early human cardiac development. More recently, techniques like CRISPR have allowed the introduction of single base pair variants, again, analogous to human variation, and can show defects as shown on the right here with abnormally small atrioventricular valves, and as well as excess valve tissue, showing dysregulation of sort of valve morphogenesis by this variant. And again, there's many groups that are using zebrafish to assess cardiovascular development. And there's also a number of labs now doing high throughput zebrafish screens, both for drug discovery and to look at combinatorial genetic effects. Of course, everybody, when we think of model organisms, we think of mice, and so the mouse and human cardiac development are very similar. There's a really elegant paper by Benoit Bruneau that goes through sort of state of the art as of 12 years ago. Is still widely referenced today. And we can really do all of the phenotyping that we can do in humans or large animals in mice. We can MRI them as shown in panel A here. We can, of course, do necropsy, which is harder in humans. We can echo them. We can do electrophysiology studies. We can do coronary anatomy studies. And we can also do several different surgical procedures. We can monitor them either transiently or over time, and mice continue to be the workhorse for cardiovascular biology. There's a few cool, at least in my opinion, large-scale mouse experiments worth knowing about. So one is a human mouse enhancer browser. It's a project by several investigators at Lawrence Berkeley National Laboratory, and has mapped over 3,000 mouse enhancers, and there's whole mounts you can look at sort of from your web browser. Jake Lucis down at UCLA has done this amazing project, taking 100 different strains of mice and doing quantitative phenotyping of them, and has shown in a number of really amazing papers to be able to do fine mapping and also look at gene environment interactions across this diverse group of mouse strains. And then we'll hear later today from the knockout mouse phenotyping projects, and there are many others, both stateside and across the world, trying to understand the full diversity of genes in the mouse model. And then finally, one of the things that being in the translational space is important, particularly for our FDA colleagues, is large animal models. And so recently, a couple of groups have successfully knocked in mutations into domestic and mini pigs, and do see in certain diseases like hypertrophic cardiomyopathy, where mouse models don't recapitulate certain aspects of disease, that in fact they can recapitulate exactly key components of the human disease, and thus are ideal models for doing advanced therapeutic development. So with that, it's by no means a comprehensive view, but a taste of what's available. And we'll take questions after.
Yes, thanks, Matt. That's great. Next, we have Wendy Chung, who's going to talk on congenital diaphragmatic hernias. And like Matt said, we can take questions afterwards. Thank you so much, Matt.
And let me just get this full size. So I'm going to take a slightly different approach. For some of you who know me, I'm a human geneticist, and I'm going to represent some of the work of my colleagues, but and I'll try and make sure I give them credit as we go through. So I'm going to talk about congenital diaphragmatic hernia. I'll just say very briefly, a developmental disorder of the diaphragm or the breathing muscle, and importantly separates the thorax from the abdomen. And thankfully, it's not very common, but it is very deadly. And one of the things that happens during development is without this separation, the intestines migrates up into the chest for that developing fetus and compresses the lungs. And a lot of the ultimate mortality is due to pulmonary insufficiency. And I'll explain why that's important in a second.
And we can't see your screen for some reason.
Oh my goodness.
We're not seeing it.
It says you've started sharing, but we don't actually see it.
Okay, I'm sorry about that. Let me try again. Thank you for saying that. Are you seeing it now?
Yes, we are. Thanks.
Okay, very good. So I apologize. Anyway, I'll just show you the picture since you missed it. But anyway, here's the problem with the diaphragm and the guts up in the chest. And we've had the opportunity to do quite a bit of sequencing of our cohort through Gabriella Miller Kids First and other NIH funding. And this has been enormously helpful, but also shown us the complexity of the condition. And so the cohort I'll be describing, we now have about 1,500 individuals with CDH, and we're in the process of analyzing all the sequence. But we know that the genomic architecture is in part due to de novo mutations, in part because this is such a lethal condition that affects fitness. And also we're realizing that there are also inherited variants that I'll get to in a second. The challenge is, though, and I'll get to the punchline, is that although this might be developmentally look like a single defect, so to speak, genetically it's quite heterogeneous. We can predict that there are going to be at least over 100 different genetic disorders that all end up being called CDH. And statistically, when we look at all of this together, and I don't care that you understand all of this, the point is that there's signal, and you can see that from the p-values on the right, but the signal is weak. And so there's a lot of noise in the background, and ultimately trying to figure out which of these are true CDH genes and which are not becomes the challenge. And so with this, and the data I'm going to show you just in general today are unpublished, but what we've been able to see, because we've just gotten to appropriate sample sizes, we have our first gene that is genome-wide significant. But the big problem we've had in the field is that we've had not enough power to be able to see genome-wide significance. And so the punchline is the animal models are extremely helpful for us to be able to differentiate what our gut feeling tells us really is a CDH gene and to get functional data to validate that. The one gene I showed you about LONP1, we see both de novo and inherited pedigrees. And I won't go into too much detail, but one of the things we've clinically seen is that this is one of our most severe associations genetically. And so a la precision medicine or trying to understand prognosis, our individuals that have LONP1 mutations have about a 62% mortality, which is extremely high even for CDH. And so part of it has been to try and understand why we have that. One of the things that's been valuable with the animal models, and in particular, I'm going to talk exclusively about mouse models, but is the ability to actually separate where the mutation is expressed. And so this is work from Jin Sun's lab. She's done a series of experiments, she and others who I'll describe. And in this particular mouse model, we've knocked out the gene, but exclusively within the lungs. To try and answer this question, was it simply a mechanical issue during development with compression on the lungs, or was LONP1 actually affecting primarily development of the lungs? And the short story is, by still having the diaphragm intact, being able to see LONP1 locked out specifically in the lungs, we can see on the left are controls and on the right, the LONP1 lungs, you can see very, very odd and cystic lung formation. And then if you look in terms of microscopy, you can see that there's branching morphogenesis defects. And so you end up with this very aberrant lung and, not surprising, problems with oxygenation and probably a large part of our mortality that we're seeing. So that's great that we can do that for a gene that has genome-wide significance, but we've also been trying to see the signal from the noise from the others. So these are genes that I'm showing you here that do have recurrent mutations within the same gene, so we prioritize them. One of those genes, NYRF, is a gene that we subsequently wanted to look at. We weren't sure at the beginning because it's named myelin regulatory factor, and so it wasn't intuitively obvious that that should be a CDH gene. But again, in terms of making the mouse model to do that, I don't know if you can see this well enough, but here's the control, nice diaphragm. Now with the knockout, you have the liver in the chest and compressed lungs and abnormal lung development. And again, this is also work from Jin Sun, abnormal lungs here and here. And I'm sorry, I'm going quickly, but it's just meant to be to show you that there are differences. And then we can see fibrosis at the periphery of the lung. And again, similarly, problems with oxygenation. We can go down to genes where we only see one person in our cohort with this defect. And so we've seen that with the gene CDC42BPB. We've used Gene Matcher, which some of you know as human geneticists, to try and see if there were other humans with this. And we did find a second case, and we're able to work with Steve Murray and his team in COMP to then model that in terms of the mouse models. And in fact, as I said, able to demonstrate within the mice as well. This is a systemic disorder in terms of not just solely associated with a diaphragmatic defect, but that also was something that we saw in humans. So we weren't surprised to be able to see sort of a syndromic or a complex phenotype as we saw that. The mice have also been helpful because there are some differences in terms of gene dosage. These are all genes where we see heterozygotes in human, and we can be able to model these either as homozygotes in the mouse or heterozygotes. And you can see the mice are similar to humans in that we often find incomplete penetrance. You can see the diaphragm here, you can see the bulging here. Again, see a subtle defect here, and then some cases where you see no defect. So good model in terms of what we've been able to see for that. And then the final case I'll briefly summarize is SYN3A, another transcription factor. And this is work that David McCulley has done with us, and really beautiful because now we can do cell-specific expression to understand within the diaphragm which cells are relevant. So when we knock it out in the fibroblast, everything looks normal. On the other hand, if you knock it out in the skeletal muscle, you see the lack of skeletal muscle in the diaphragm. And if you knock it out only in the mesothelium and crural muscle, you see exactly the type of diaphragmatic defect that we see. And again, being able to see that in the lung, seeing a specific lung phenotype, not simply due to the compression as well. So just briefly in conclusion, and I'm sorry it was relatively rapid, but being able to do this, we can see that there's definitely genotype-phenotype correlations that we'll be able to, I hope, use clinically by understanding the cells that are relevant and the phenotypes that are relevant, hopefully be able to think about ultimately what we might need to target molecularly. And finally, just to say it takes a huge team of people and to specifically call out those who I mentioned earlier. So thanks, I'll stop there.
Wonderful, thank you, Wendy. Next up, we have Elizabeth Engel, who's going to talk about cranial disinnervation disorders. We see your screen.
Sorry, I was muted. And you can't do Zoom and mute at the same time. There we go. Now can you see it and hear me?
Yes, yes, Debbie.
Great, thanks. Similar to Wendy's beautiful presentation, my lab studies sort of a domain expertise in human developmental disorders that result from errors in cranial motor neuron development, and in particular, those that result in absent or abnormal eye and face movements. And as a group, these disorders are referred to as the congenital cranial disinnervation disorders, or CCDDs. Many of them are inherited, and this allows us to use genetics to probe their etiology. Eye movements are controlled by connections from the brainstem to the extraocular muscles by the abducens, trochlear, and ocular motor nuclei and nerves. And these nuclei are anatomically well-defined, and they innervate the muscles in a very precise and tractable fashion. Similarly, the larger facial nucleus contains 4 bilaterally paired subnuclei, which also innervate multiple facial muscles. We're very fortunate because the cranial motor neurons are evolutionarily old, and they're conserved in both mouse and fish, as well as other organisms, allowing us to successfully model CCDDs in a variety of organisms. Moreover, we can visualize these developing cranial nerves beautifully in both mouse and fish using genetic reporters. This mouse at E15.5 has nerves in green and arteries in red. Thus, we approach the CCDDs in a very similar way to others who are on this call. We identify the spectrum of human disorders, try to define their genetic etiologies, study their developmental pathology and disease mechanism, as well as the role of the gene in normal development. And this in turn hopefully informs both neurodevelopment and patient care. So using this approach, we've defined a series of Mendelian genes that when mutated cause very specific forms of CCDDs. I just want to highlight here the orange, what I've highlighted in orange are the genes that work through a loss of function mechanism. And we're fortunate that knockout mouse models for many of these were already reported, and the mouse phenotypes corresponded nicely with the human phenotypes. We imported a subset of these for more detailed mechanistic studies. The 3 genes in red, KIF21A and TUB3 that I'm going to return to in a moment, and CHN1 cause CCDDs through missense variants that alter protein function, so they're much more specific. And in each case, we've introduced the missense change 1 or 2 or 3 into mouse and recapitulated the human phenotypes. So using such models, we found that these mutations perturb different stages of neuronal development, not surprisingly. The first group includes mutations that alter motor neuron identity or differentiation, and as a result, the motor neuron dies or it's respecified to another identity, and the axons can't innervate the target muscles. The second group of gene mutations cause errors in growth and guidance of cranial axons. But despite these successes, a very large % of our cohort remains unsolved genetically. And so we're really fortunate that through the Gabriella Miller Kids First Initiative, as well as the Broad Center for Mendelian Genomics, we have whole genome sequencing on about 1,000 individuals with a variety of unsolved CCDDs. We partnered with the Broad to harmonize these with reference samples in NOMAD database and for alignment and variant calling, and my lab is now interpreting those variants. But one of the biggest challenges is to prove pathogenicity and to choose which variants to pursue mechanistically. To support pathogenicity, as Wendy mentioned, we search for additional alleles, we interrogate our in-house cranial motor neuron functional genomic data, and we perform targeted in vitro assays. One of the things we lacked, however, was a moderate in vivo anatomic screening mechanism to support pathogenicity. And so for this, Julie in the lab has turned to zebrafish. We have live fish that harbor reporters for multiple cranial nerves and muscles, as seen on the left, or ones that are more specific. For example, this one for abducens motor neurons and axons on the right. And these can be imaged to determine if variants alter cranial nerve development. Julie decided to test a recently published rapid method for directed gene knockout in G0 fish. This is based on simultaneous high-dose injection of 4 guide RNAs, redundantly targeting a single gene, and was reported to produce homozygous null phenotypes in greater than 90% of the embryos without very great toxicity. Julie has successfully reproduced these results, and she can now screen for a phenotype in about 72 hours, which would take months to generate in germline fish. She first tested whether she could recapitulate known CCDDs that result from loss of function variants. And here we'd reported a long time ago that loss of function variants in the transcription factor FOX2A causes a disorder called CFUM type 2. And we and others found that with loss of FOX2A in mice, or as you see here in germline fish, you find absence of the ocular motor and trochlear nuclei and nerves. Note that on the wild type circle on the left, the circle contains the 2 nuclei, while the dashed circle on the right is empty. And so Julie successfully recapitulated these germline findings at 72 hours using the injected pooled guides. After confirming several additional known CCD genes similar to this, she now has a smooth workflow for her G0 screen. She's tested about 71 genes for loss of function with preliminary G0 pilot injections, and 38 genes have putative appropriate craniomotor neuron phenotypes. The F0s for most of these are now adults and being crossed to examine the developing embryos. And I'm just presenting this because we're hopeful that this may be a feasible approach to screen at least loss of function candidates in a rapid way, not only for CCDDs, but for other developmental phenotypes that may be visible in zebrafish. While Julie's working on fish, though, similar to Wendy, our lab primarily models in mouse, and we're really grateful for Comp2 for our collaboration with Kids First and Comp2 that's made several new mouse models that I just wanted to briefly introduce to you. For the first one, I need to tell you a little bit about these forms of congenital fibrosis syndrome, or CFUM, resulting from KIF21A and TUB3 recurrent missense changes. And I hope this isn't too confusing, but this child has an isolated eye movement disorder, CFUM type 1. We found this disorder to result from recurrent missense variants in KIF21A in very specific regions. And KIF21A encodes an anterograde kinesin motor protein that you see here walking along a microtubule. We found that mice harboring this child's KIF21A variant do have CFUM, and it arises from an axon growth disorder in which a subset of axons stall between the brainstem and orbit. We also found mechanistically that when inactive, KIF21A maintains an autoinhibited conformation, so it can't interact with microtubules. And when we introduce these mutations, we attenuate this autoinhibition such there are too many KIF21A molecules running up and down the microtubule at any given time. By contrast, this child has an often indistinguishable form of CFUM, but it is syndromic, and it results from specific recurrent missense mutations in the gene TUB3. TUB3 encodes a tubulin isotype that's a building block for the neuronal microtubule cytoskeleton, and that's a cytoskeleton upon which this kinesin is walking. In addition to CFUM, he has dysfunction of additional cranial nerves, he has intellectual and social disabilities, and a progressive axonal polyneuropathy. We found that mice harboring his variant have a more severe misguidance of cranial axons as well as other central axons. And these TUB3 variants actually reduce the association of KIF21A with microtubules, the opposite effect of the recurrent missense variants in KIF21A. Thus, we've been trying to understand the mechanistic relationship that there may be between these two disorders. And this is where Comp2 comes in. Much to our delight, we identified a novel KIF21A missense variant, so a KIF21A missense variant that causes the identical syndromic TUB3 phenotype. And so we're thrilled that Jax has made this mouse for us, led by Steve Murray, and we've recently imported it and hope that it will help us to understand the mechanistic link between these two disorders. We've also identified specific TUB-A1A missense variants. That's a new genetic cause of CFUM, and the Comp2 UC Davis team is generating this mouse. It's quite difficult to make because of homology, and they've tried multiple attempts to generate it. So in closing, I just want to highlight, I hope this highlighted how we use both mice and zebrafish. And I want to especially acknowledge and thank the Kids First and Come2 programs and the collaborators who've worked really hard to generate these new mouse lines for us. Thank you.
Wonderful. Thank you, Dr. Engel. Up next, we have John Maris, who's going to be talking about PDX neuroblastoma mouse models.
Thank you very much for the invitation to be here today. A very interesting talk so far. So yes, this will be a little bit different. I'm a physician scientist at the Children's Hospital of Philadelphia who is focused on childhood cancers, but mainly this disease, neuroblastoma, and that's what our Gabriella Miller project is. Just very briefly, childhood cancer is thankfully rare, but this is one of the more common childhood cancers and the most common malignancy that happens in infancy or diagnosed in utero. Like all childhood cancers, it's a developmental defect. So it's a defect of the sympathetic nerve system as the cells migrate out of the neural crest. It is this is the prevalence, about 1 in 7,000 births, and it's an important childhood cancer as the clinical courses can be quite diverse, with some having significant amplification of a gene called McN, which is all of these red dots, extra copies of McN in the nucleus of a neuroblastoma cell, which makes a very, very aggressive phenotype. Yet other children have a phenotype of spontaneous regression. So that complexity has always been both fascinating and also frustrating as a clinician. So our XO1 cohort, it came from a study done by Andy Olshan, where he was looking at environmental factors that influence neuroblastoma. And the reason why this was important is that while we do a lot of banking of neuroblastomas and blood and other samples from the children with the disease, we as a community have not been routinely collecting blood samples from family members. And that's why we focus on this study, because dyads and triads were available, and a large part of this is focused on germline aberrations that lead to neuroblastoma. So unlike many of the Gabby projects, in addition to the germline, we have a lot of tumor DNA and RNA sequencing as part of this project. And that's where the patient-derived xenografts come in. While there is a role for model organisms to focus on the germline variants, my talk here today is really on the somatic variants and what we're doing about that in terms of functional and drug development activities. So just very briefly on the germline, this is what we have found in the cohort, that 13% of the patients in our cohort had a pathogenic or likely pathogenic mutation in these genes that you see here, which are all bona fide cancer predisposition genes. And so the other very important finding that was somewhat surprising, at least to me, in that in all but 2 cases, these germline variants were inherited from one of their parents. And the big focus of our work is now overlaying polygenic risk scores to understand how these Mendelian variants are influenced by common variation and why the kids got the disease and not their parents. But what we're focused on here today is this landscape of somatic alteration. So this is not from the Gabby project. This is a paper that we published in collaboration with the St. Jude group last year. And what this is showing you is that there are age-specific chromosomal aberrations and single nucleotide variations that vary in their frequency based upon age and the stage of the disease, et cetera. And while this is complex enough, these data were all generated from tumors taken at the time of diagnosis. And what happens to all patients after they get diagnosed is that we expose them to very genotoxic chemotherapy and radiation therapy. And so again, outside of the Gabi project, we have multiple different efforts to try to understand the genomic evolution of this disease. And this is work from Esther Burko and Yael Mosé, where we have multiple samples. So this is at diagnosis and then at relapse and at multiple relapses. And what this fish pod is showing is the mutations that were present. So there are at least 3 clones were present at diagnosis, and how the major clone went away, but then reappeared, but multiple other clones. So this is a very complex disease, and it is somewhat humbling to try to figure out how we're going to take this sort of underlying complexity and come up with curative therapies. So patient-derived xenografts are human tumor that comes out of the patient. It's directly implanted into a severely immunocompromised mouse. The engraftment probability directly relates to tumor aggressiveness. And as much as we would love to study the spontaneous regression phenotype, we can't get xenografts from those sort of patients. Many people, including our group, have shown that these tumors are a faithful recapitulation of what's in the patient by not going into plastic and hyperoxia. They maintain many of the tumor cell intrinsic features of what we see in patients. And therefore, they serve as very good models for those sorts of features. But because they're in an immunocompromised mouse, the tumor microenvironment is, of course, completely artificial, and these are not good models for development of immunotherapies. So we published, with the support of Alex's Lemonade Stand Foundation, a paper as a resource, which was really just taking our patient-derived xenograft models and throwing every genomic technology that we could get our hands on at them to provide a landscape of what these are for drug development purposes. And so we had, in this effort, we had 261 patient-derived xenograft models. You can see they're from a lot of different childhood cancer diseases, a lot of different genomic technologies to annotate these. And to make a long story short, this proved to us what we needed to know, is that these characterized models are a good reflection of relapse disease. And so just to focus on neuroblastoma, since that's the topic here today, we had 12, in this effort, we had 12 patient-derived xenografts established from diagnosis, 23 from relapse. And as we have proven in the past, that the frequency of ALK mutations are much higher at relapse because many subclones present at diagnosis cause drug resistance, and the frequency is higher at relapse. And so for all of these diseases, we have a very good characterization of the genomics of this disease, and this is very useful to us, as I'll show you in a minute. And also on the RNA side of things, it was very, it was reassuring to know that, you know, all the rhabdomyosarcomas clustered together, the osteosarcomas clustered together, all of the leukemias clustered together, all of the brain tumors clustered together, and that the many childhood cancers are driven by fusion events, chromosomal translocations to create oncoproteins. Others are not, and our data, again, capture all of that. And, you know, why did we go through that effort? Well, to develop drugs in a smart way, you need to know these data. So this is, these are data, again, from Yael Mosé, where this kinase ALK is the most commonly mutated oncogene in neuroblastoma. It's druggable. There are kinase inhibitors. And as you can see in this patient-derived xenograft model, that you can measure it here at the time of study entry. If you don't do anything or treat with a placebo, it grows quite rapidly over the course of several weeks. This drug here, crizotinib, worked a little bit, but not great. Whereas this drug, which had just a number at the time, now known as lorlatinib, caused complete sustained regression in both of these models. And crizotinib was ahead of lorlatinib in terms of development. It entered clinical trials before, but based upon these data, we have replaced lorlatinib for crizotinib, and it's now being tested in a phase 3 upfront clinical trial. And then on the RNA side of things, there's a lot of interest in cancer, childhood cancer, on developing new immunotherapies. And having an army of these patient-derived xenografts allows us to test hypotheses, such as this drug, which is an antibody has a toxin attached to it against a protein that we know is differentially expressed in neuroblastomas. Most have very high expression, but some have low expression. And in this study, we were able to show that this cell, based on the RNA and the protein, really had no expression of DLK1, and the drug didn't work at all. This one had a little bit of expression, had a little bit of activity, but all of these tumors down here had very high expression and very good anti-tumor activity. So patient-derived xenografts are just one type of model for neuroblastoma, and there are many others, and I'm not going to read through this list, but a model is only as good as your knowledge of its strengths and weaknesses and the hypotheses you're testing. We find patient-derived xenograft models very useful for drug development purposes and other functional validation work, but a lot of the hypotheses that are coming out of the Gabriela Miller project as a whole will be tested in other model systems. And so I want to thank everyone who's been involved in the Gabi project, this pediatric preclinical testing consortium where I showed data from, which is an NCI-funded effort, as well as our funding sources and people in my lab. So thank you for your attention.
Thanks, Dr. Maris. Let's take a few minutes to see if anyone has any questions.
I'll ask questions. Oh, go ahead.
I just had just a question for Wendy, which is, did you anticipate that that these genes would have selective expression alterations in the lung as well? And do they tend to be ubiquitously expressed and for some reason are hitting certain tissues? And it's interesting that those with primary lung effect also obviously have to have primary diaphragm effect, right? So like, why the association, and is it based on gene expression or something else, do you think?
Yep, so the ones that we've looked at so far, we actually prioritized the genes that we thought we would try in mice, for instance, based on expression, tissue expression, and thinking that lung and diaphragm, and to be honest, also heart development are sort of coordinated and have some similarities and overlap. So I think it's a winner's curse, so to, in a way, in the sense that we kind of pick genes that would happen like that. If you'd asked me 15 years ago when we started studying CDH, though, I naively thought that most of the pulmonary stuff was simply a mechanical issue developmentally.
Yeah, as pediatricians, that's what we were always led to believe, right? So, yeah.
So I've gotten smarter with some experience. So, but I think it's probably going to be a lot of the lung pathology with the very, very severe outcomes is going to be primary lung problems.
So are there equivalent disorders that don't have CHD because they only don't have diaphragmatic hernias because they only affect the lung in a similar way?
So the interesting thing is when you start looking in a genetics first approach, right? So now if you take the gene and look at the phenotype, for some of those genes, we only see CDH in about 10% of mutation carriers. And what's most consistent and highly penetrant for many of those genes are actually the brain manifestations, interestingly enough. That may be the most sensitive in terms of dysfunction. And now that we know what we're doing, we're starting to do more specific PFTs and imaging and trying to get what may be subclinical, at least for young children, be they pulmonary manifestations or in some cases even other systems that we're starting to look at now. So the mice are sort of pointing us at which organs to pay attention to.
It's very cool and very beautiful. Thank you.
Dr. Engel, there's actually a question for you, or not for you. It was about the fact that you talked about using zebrafish for moderate throughput, and Danielle Day is asking, is that what some of the other researchers who are here today are using as well?
I'm sure others are trying this because it was published in 2018, and I don't know though if anyone else I don't know of anyone else testing it right now, but we haven't talked about it widely. So
We do have Yvonne back.
Yeah.
On the, she's going to be on the panel later, so maybe she could speak about if she knows about any studies that are doing these high throughput screening models in their models. I think this brings us to our break. So unless there's any other questions that anyone wants to ask.
I'd like to just put a bug in folks' ear for our discussion later while we go off to break, if that's okay. I think, what was really spectacular about all this wonderful science was just seeing the diversity of phenotypic features that were discussed and shown in all kinds of different ways. And I guess I'd like to encourage you to think about how would you actually look at some of those in a cross-species manner? How would you validate it in other species to examine portions of the mechanism for particular reasons in other species? And I think one of the things that we really want to encourage in the Kids First and Monarch collaboration is increased computability and relationships across species. So, obviously we choose the organisms that we choose for a variety of reasons, but anything we can do to support that sort of cross-species inquiry is really helpful to other investigators.
Great, thanks, Melissa. So yeah, let's take a 5-minute break. Let's resume at 5 minutes past the hour, and we're going to come back with a brief keynote from Damien Smedley, who will talk about the diagnostic successes in genomics.
Yep, no, that's perfect now. I think, Nicole, whenever you're
All right, shall we get started again? Welcome back, thanks everyone. And to kick off the next part of the seminar and the webinar, we'll be having Damien Smedley give a brief keynote on diagnostic successes in the 100,000 Genomes Project, followed by a panel discussion with several speakers. So Damien, please, thank you. Get started.
Okay, thanks a lot, Nicole, and welcome everyone. So I'm going to talk about how we use phenotypic data for rare disease diagnosis in the 100,000 Genomes Project, but I'm actually probably going to more focus on the lack of success, like the 75% of cases where we don't have a diagnosis after whole genome sequence, and how model organism data can hopefully help us in the future. And that will lead on to the panel discussion. Okay, so just to give you a brief bit of background, so the project was launched back in 2014 by the UK government and NHS England, and it actually completed sequencing at the end of 2018. And now pretty much all the reports have gone back to the NHS. In terms of mission, so this is kind of fairly obvious, but just to sort of make it clear, obviously to bring benefit to the patients from sequencing over 100,000 whole genomes from participants with both rare inherited disease as well as cancer, but also to enable biomedical research. So all the data is put in a research environment, a bit like Kids First, to stimulate the UK genomics industry particularly, and obviously to do this all in an ethical and transparent manner. And so this slide's even a bit out of date now, but actually we ended up with about 120,000 genomes sequenced, and the majority of them, so over 80,000, come from rare disease participants or their family members. It was actually easier to kind of recruit rare disease people than cancer, believe it or not, for various sort of technical reasons. And this is the part that's more out of date. So we've actually returned results back to the NHS genomic medicine centres for nearly all the samples now. And in terms of how we use phenotype data, so we'll probably get into ontologies later. So like phenotypes were collected for all the rare disease patients and using the human phenotype ontology. And we used a tool developed in my team and Peter Robinson's team and various other members of the Monarch Initiative called Exomizer that attempts to start off with the whole exome or the whole genome sequence and the family information. And it kind of does the obvious stuff of kind of removing non-coding variants and removing common variants, looking for segregating variants and trying to prioritize by like how rare and predicted pathogenic those variants are. But on the other side, it kind of automates phenotypic comparisons between the patient's phenotypes and reference genotype to phenotype knowledge, either coming from databases such as OMIM and Orphanet and those HPO annotations, or from the model organisms that we're going to hear a lot more about today. So in Exomiser, we're using mouse models from MGI and the IMPC, and zebrafish models from ZFin. And we also use protein-protein interactions to kind of fill in the gaps. And the idea of this is that, so rather than just using sort of genomic data to try and come up with like a single variant diagnosis for a patient and hopefully some potential treatment, we want to bring in like underutilized data on the patient's phenotypes, and eventually sort of going into other omics technology and environmental data. So these are future aspirations. And we want to, mnemonic, but hopefully in Exomiser eventually we want to use as many different species as possible. So obviously I've just said we're only using the mouse and zebrafish at the moment. I don't know why my slides keep changing, apologies, but in the actual mnemonic knowledge base, we have data on a lot of other organisms, not just models, and we'd like to bring that into this approach of Exomiser eventually. And we've talked a bit about ontologies and comparisons, but essentially in all our tools, such as Exomiser, we're comparing a patient's phenotypic profile, shown here in the middle, with reference genotype to phenotype knowledge from human diseases over here on the left. So if we know a particular gene is associated with this disease, and we can show it's phenotypically similar using our algorithms to the patient's phenotypes, then we know that a variant in that gene is a good candidate. And similarly, we can automatically compare to mouse phenotype models curated from the literature, either by MGI or from the IMPC comp database. And then coming back to our results on the 100,000 genomes project. So we put together a pilot study of the first sort of 2,183 cases. So this is 5,000 genomes in total because we've sequenced family members. And this is like the big study where we've kind of collected all the data we can possibly put together. And as you can see, like up here at the top, the actual diagnostic yield is 25%, which may seem quite low compared to some other projects. It does vary depending on the family structure. Obviously, you're recruiting larger family structures, you can do more filtering. For the categories that are more likely to be monogenic, the diagnostic yields up at around 35%. And in this bottom panel, you can see the diagnostic yield varies a lot by disease. So yeah, for intellectual disability and eye diseases and hearing diseases and other neurological diseases, we can get diagnostic yields above 40%, as you'd expect. But as part of the 100,000 genomes project, we were kind of taking all comers in our national health system. So we had a lot of participants suffering from Turner syndromes, which would just turn out to be much harder to diagnose. Yeah, the yield was down to 5% for these. But phenotypic data helped us a lot. So here I'm kind of talking about the sort of first pass diagnosis, and this is mainly based on known sort of human disease genes. The blue bars here show the recall of these diagnoses. And the other thing I've not mentioned yet is Genomics England and the 100,000 Genomes Project, as well as using Exomiser, they use a virtual panel-based approach. So they decide for a particular participant based on their recruited disease and phenotypes. They apply sort of virtual gene panels to sort of narrow down the search. So that approach using panels recalled 77% of the diagnoses. And then, but using the sort of phenotype-based approach of Exomiser, we could recall like 77% as the top ranked hit using Exomiser. And if you sort of go out to the top 3 or the top 5, you can improve that further to recall some sort of 86, 87% of the diagnosis. And the sort of panel-based or Exomiser approaches are kind of complementary, so putting them together is even better. Yeah, I won't sort of focus on why we sort of missed the sort of 10% we don't, but basically what I wanted to end on and sort of lead to the panel discussion is this is all great. You know, we can use phenotype data to really precisely recall those diagnoses, but still like for 75% of the patients, we're not managing to come up with a diagnosis or help them. So what can we do about these cases sort of going forward in the future? So yeah, the lowest hanging fruit is going to be kind of overlooked sort of coding SMVs or indels in known disease genes. So we can go back and look at our exomiser candidates in a bit more detail. Yeah, a lot of the NHS clinicians have kind of focused on those applied panel genes, but there might be some more obvious diagnoses outside those panels. Obviously, reinterpretation, running this pipeline every few months, there's always new knowledge. There's other software, so Lyrical was published last year by Peter Robinson. He sort of led on this, as well as other people from the Monarch Initiative. But as well as looking at the sort of coding SMVs, yeah, staying in the variant space, we want to sort of start looking more at non-coding diagnosis. So in that gel pilot study I was talking about, actually 4% of our diagnosis came from non-coding variants in the end. So these are either sort of known pathogenic variants that you can find in ClinVar, or novel variants that we managed to validate through research analysis and identify using tools such as Genomizer. And particularly at the moment, people are starting to really focus on the cryptic splice variants. So using tools such as Splice AI, or a new sort of tool called Squirrels that's been developed by Monarch. Another type of variant, structural variants, are going to account for like a good 5 to 10% of diagnosis, we hope. So we're starting looking at these. So the next release of Exomizer, we're going to do structural variant as well as SMV prioritization. But sort of coming back to the theme of this session, so hopefully a sort of good proportion are going to be diagnosed by sort of new disease gene discovery. So variants and genes that are not human disease genes at the moment, or not known to be human disease genes at the moment. And here is where hopefully we can use some of the exomizer candidates based on model organism phenotypes or protein-protein interactions. So we've already started running this pipeline, and we've sort of shown that we've got hundreds of candidates identified in our unsolved case. So these have been quite conservative, just looking at de novo or loss of function variants in novel genes, where the phenotypic evidence is either coming from mouse models or from like protein-protein interaction, guilt by association type data. And I'll also sort of show you another approach where a PhD student is in my team that has been screening novel hearing genes identified by the COMP-IMPC pipeline. And another thing we've been doing in my team is to do burden testing to detect enrichment of rare segregating predicted pathogenic variants in novel genes, where we're sort of looking at disease cases versus controls. And we've got some 977 novel associations to try and triage. And here again, the sort of model organism data is going to help us functionally validate those. So just to sort of end with some examples. So first, using the latter, this sort of burden testing approach. So hereditary spastic paraplegia and UBAP1. So we managed to detect rare loss of function variants using Exomizer in some 5 out of 417 cases versus only 3 controls. And here there was like some 16,000 controls. And the case variants were completely absent from the controls or NOMAD. It looks like a haploinsufficient gene from NOMAD data based on the sort of observed expected ratios for predicted loss of function variants. And actually, when you go back and look at the Exomizer data, this one was based on protein-protein interactions with another known spastic paraplegia gene. And actually, as soon as we identified this, another sort of group published a confirming study in the American Journal of Human Genetics. I think I'm sort of going over my time a bit, so I'll skip this example and sort of just talk about some of the mouse model examples that candidates we've pulled out. So just a couple. So intellectual disability in this PDS5B gene. So we've got a case here where we've got a de novo variant because we've sequenced the parents as well. So it's rare, predicted damaging missense variant. And it was the top ranked Exomizer candidate based on a mouse model where the microcephaly, atrial septal defects, bone ossification, and short limbs were matched. So this is a kind of variant that we'll want to go back and look for other confirming cases, independent cases. Another example, this ultra rare undescribed monogenic disorder recruited category, where we found a rare frameshift elongation de novo variant, again from sort of trio sequencing And here, like the mouse models providing evidence of liver abnormalities that match the patient's phenotypes. And again, the sort of nomad haploinsufficiency data matches nicely. And the final one where the mouse models matching a syndromic cleft lip palate sort of patient phenotype. And then this is the work I was talking about earlier. So this is the work of Leticia, who's sort of a shared sort of joint PhD student in my team at UCL. So what she's done is, a few years ago, there was a IMPC paper where they did a large-scale hearing loss screen and identified some 67 candidate hearing loss genes, and 52 of these were novel, so they've never been reported before in humans. And there's also been a GWAS recently published last year, or a couple of years ago now actually, in using UK Biobank data. So what Leticia has done is to look at all the 100,000 Genomes Project unsolved cases and just look for kind of rare protein altering or protein truncating variants that segregate in either the genes from the IMP study or the GWAS. And sure enough, she's got some nice sort of candidates in some 80 genes where the patients are annotated with hearing impairment HPO terms. So we sort of going through and validating some more of those. And I just wanted to finish. So we've obviously done this for the 100,000 Genomes Project, which is delivered by Genomics England, and sort of delivered this as part of an ISA accredited service. And yeah, we kind of realized that Exomize is kind of open source software that you can sort of freely use and download and install yourself. But for some users, they want like an easier to use solution. So we've been working with Congenica to integrate Exomizer into their decision support tool, and also to develop a sort of standalone, easy to use version of Exomizer. With that, yeah, it's one of these projects, the 100,000 Genomes Project, plus all my colleagues in the IMPC and MONIC initiative. There's kind of too many names to fit on the slide, but mainly to sort of thank the patients and their families who took part in the project. And with that, I guess I can answer questions as part of the panel discussion, but I think we need to move on to the panel discussion. And sort of prior to the meeting, we kind of asked the sort of 4 people on the panel a set of 3 questions. So I'd like to invite them to kind of like give answers to these first questions, the first question. So starting with question 1, like what data does your database host and how can researchers find or use it? So for the panel members, like feel free if you want to share your screen, if you feel that makes it easier, but I guess I'll go around each of you in turn to sort of simplify things. And then if other people on the meeting have got questions, they should jump in as well. You'll have to kind of help me sort of spot the questions coming up. But should we start with Pilar, sort of representing the IMPC mouse models? Do you want to kind of give your thoughts around question 1?
Yeah, sure. Thank you, Damien. So, I mean, to be honest, I was very pleased to hear already through some of the talks during the first session about some successful stories using comp mouse. So I heard some successful collaborations with Steve Murray, with Ken Joy, that are part of the IMPC. But these were cases where they were focused on specific genes or specific targeted mutations of interest. But other than that, the IMPC has available a website, a database that I don't know if maybe I can, I mean, I guess most of you are familiar with it already, but maybe I just can share my screen. I don't know if that might be useful.
Yeah, I guess we got a minute or 2.
Okay. Well, very briefly, this is the IMPC website. So just to summarize, our database hosts phenotypic data for knockout mice genes. And here you have the total number of genes that have already been phenotyped, and we have data available, so over 7,000 genes right now. So we have very frequent data releases, several data releases every year. So the data is updated very, very frequently. This is the latest one from last month. And just a few things that I would like to highlight for those of you who are not that familiar with this resource. It's like this is data that we are generating. So we are generating the knockouts and we phenotype the mice. So it's not data aggregated from the literature. We are targeting genes that are yet to be studied. So genes for which we don't have many information about the function, what we could call dark genome. So we are targeting particularly those genes. And then that we have these standardized phenotyping protocols that we apply both to the wild type and the mutant mouse. So we apply the same standardized phenotyping protocols, and then through a series of statistical tests, we are able to assess the significance of the abnormal phenotypes that we observe. So, I mean, just a very brief summary of what the IMPC website or catalog might offer.
Okay, thanks, Pilar. I guess we can move on to Todd now, if you're ready. Sort of
Yes.
describe the canine models.
Yeah, and I think I will briefly share my screen as well, just since this may not be something that people are familiar with. So I work on the Integrated Canine Data Commons. This is part of NCI's initiative called the Cancer Research Data Commons, where data from various cancer projects are stored in cloud-based systems, and users can get access to it from there. Unlike the rest of the data repositories in NCI's initiative, the Integrated Canine Commons is actually devoted to single organisms. The others tend to be like devoted to genomics or imaging, a specific data type. But canine is devoted to research on cancer in dogs. Primarily, it is currently looking at various various clinical trials. Since dogs spontaneously generate cancer, there are clinical trials on how to treat them. And the data from some of these are showing here, just in the Canine Commons. And the basic of the idea behind this site is that it allows users to search on a variety of factors to find data that is of interest to them. They can then take that to one of NCI's cloud resources, which are three sites that are devoted to giving you workspaces, support in running analysis tools in the cloud, that sort of thing. And because it's part of the Cancer Research Data Commons, it makes it fairly straightforward to pull data both from the Canine Data Commons and from the other human-based data commons, the genomics, imaging, and proteomics data commons. So that is our basic mission.
Okay, thanks, Todd. Should we move on to Mardi next? Describe ZenBase.
Hi, yeah, so ZenBase hosts all of the genomic, biological, and expression data for the model organism Xenopus, which is used as a powerful model for human disease and congenital defects. We're a very gene-centric database, and we have over a decade of Xenopus expression data. Two features that I'd highlight that are relevant to this discussion are our recent launch of geo and support for phenotypes. So we were relatively late to start curating phenotypes compared to other model organism databases, but that gave us an advantage in that we were able to consult with other resources such as the Alliance and Monarch and other databases to get our systems to align, and that's in a way that is good for cross-species analysis. So one example of this is that we worked with Nico from UPheno, as was mentioned earlier, and I know he'll talk more about it later, but essentially it's a framework for ontologies that will allow independence of terminology heterogeneity. So it'll be really good for cross-species comparisons, especially for us, for our Xenopus phenotypes compared to humans, as well as other model organism databases. And the second feature I'd like to highlight is our support for geodata. So we generated a standardized pipeline for all the RNA-seq, ChIP-seq, and other types of sequencing data that's available at Geo. And we reprocess that all on ZenBase and support that and curate the metadata. And this allows a good source for researchers to have everything centralized and to mine that data. And in terms of phenotypes, this gives us computed gene expression phenotypes that can be good to understand the genomic basis of a disease.
Okay, great. Thanks, Marty. And then finally, Yvonne, to talk about ZFin.
Hello. I think what I'll do, I'll just share my screen just briefly of ZFin just to give you an overview of the website real fast. So ZFin is the model organism database for zebrafish research. We work on, or we display genomic, genetic, gene expression, phenotype, and disease model data. And we try and make it easily searchable through various routes and at our home site. We are participating in the Alliance of Genome Resources, where we're trying to bring together multiple species data from the various model organism databases to facilitate understanding of gene expression, phenotypes, and disease models. We've been participating in, so we curate, all of our data is expertly curated from the primary literature. So we are pulling all of our data, all of our curators read the data and insert it into the databases. We've been participating in phenotype curation from the outset. We've been doing primary literature curation for 20 plus years and are most interested in trying to facilitate gene discovery and disease model discovery and understanding how genes are involved in human diseases and phenotypic outcomes.
Yeah, actually, I had a quick question. Do you bring in data from high throughput projects? Because I remember there used to be the zebrafish mutation project, but
yes, we do. So we've brought in data from Sanger as well. I'm going to quit sharing my screen here. We brought in data from Sanger as well as there were some other high throughput screens done at the Hopkins lab. So we've brought those in and have access to the variant data as well as the phenotype, and we link out to, excuse me, Sanger and also stock centers that carry some of those strains, like the zebrafish, the ZERC, the zebrafish stock center that houses some of the Hopkins lab strains.
Okay, great. So I guess we can move on to the second question. Okay, I found it. So yeah, do you have examples of how these data informed a clinical diagnosis or resulted in a high impact genetic discovery? That's quite a high pressure question, but people might not have an answer. Let's just keep the same order. I was going to be fair and like reverse the order, but it'll just confuse things. So that we'll start with Pilar again.
Okay, so I mean, obviously there are many. I can picture many use cases for the IMPC data. Maybe the most straightforward will be to narrow down a list of candidate genes that in a sequencing study. But I mean, I would like to show you maybe some results of one successful. I think is a very successful story. One study that we have done with the IMPC data. So other than producing the data, analyzing the data, and make the data viable for the wider research community, we also analyze our own data ourselves to try to get the maximum biological insight. So apart from the specific phenotypic data across different physiological systems, the IMPC also performs a primary viability screen to assess the viability or lethality of every line. So over the last few years, working with this viability data, we know that disease genes were enriched for genes that are lethal in the mouse. So based on these previous findings, and using IMPC viability data, combining it with data essentiality from human cell lines studies, we were able to identify a specific category of lethal genes that we call developmental lethal, that are genes that are not essential for a cell to proliferate, but are essential for an organism to develop. And it was this subset of lethal genes that was driven this enrichment for Mendelian disease genes, and particularly for developmental disorder genes. So based on this finding, we apply a strategy prioritizing genes that are developmental lethal, that are highly intolerant to loss of function variation, and we came out with a candidate set of genes. So what we did next is to investigate unsolved cases from different disease sequencing consortiums, including Genomics England. So we have access through Damien to this data. We also included data publicly available from DDD and the Center for Mendelian Genomics. So we look at unsolved cases with de novo variants in this set of genes that we prioritize. And then we look at the overlap between the phenotypes observed in the patients and the phenotypes observed both in the embryo and in the adult mice. And we came up with a list of top 10 candidate genes. So this was published last year in Nature Communications, last January. And I mean, it was very, very gratifying to see that just 1 year later, 2 of these top candidate genes were already validated, and these genes have been already published, being linked to new neurodevelopmental disorders. So I think I at least I consider this a successful use and a successful story using the mouse data.
Okay, thanks, Pilar. And then, yeah, moving on to Todd.
Yeah, this is where the Canine Commons, I think, is at a little bit of disadvantage since we only opened our doors at the end of August. Most of the data we have on site is currently under embargo for publication. We hope to change that very shortly, but we simply haven't been around long enough to have much impact. Sure. Yeah, our hope is that since we're hosting the kind of data that's useful, it'll be genomics, clinical trials, cell line-based genomics, that by having it easily combinable with human data, we can support the kind of analysis that this group is talking about.
Okay, thanks.
Oh, sorry.
Yeah, go ahead.
If we have time, this is Adam from the DRC. For the maybe Todd, even in the canine context, or as when establishing these platforms, what are the workflows, or are there common workflows already established associated, let's say, with canine and canine genome analysis that are available and or can support more rapid comparative assessment at the genomic context across the canine genome and, let's say, the cancer genome, or things along those lines, if that makes sense?
Yeah, no, that actually makes perfect sense. And it's an incredibly good question, because we've actually been looking into it exactly for a little while now. Part of the governance structure of the Canine Data Commons is the Best Practices Subcommittee. This is a group of scientists who are actively involved in canine research and in human canine comparison. And right now they're developing a list of tools and processes and standard genomes that we would recommend for people to use as they get doing their analysis. Since so many people will be using canine data for such a variety of reasons, we're not trying to go much deeper than that. We're just going to leave it up to individual researchers to deal with that data as they see fit. But some best practices coming forward, I think will be very prominent in our recommendations.
Sounds great.
Okay.
Thanks.
Yeah, thanks a lot. And then over to Mardi.
So there are a lot of prominent initiatives right now that are using Xenopus as a rapid, inexpensive screen to tease out the functional role of genes that they're discovering from patient screens. And so it ranges from a variety of organ systems and disorders, from craniofacial disorders, congenital heart and kidney defects, as well as autism spectrum disorder. So a good example of that recently came out this week, they took an approach where they knocked down 10 of the highest risk autism disease in parallel using Xenopus to try to find out where all of these etiologies converge on this autism phenotype. And what they found is that it was really at forebrain growth and cortical neurogenesis. But what they were able to do follow up with that, which is good in Xenopus, is they could just do a rapid screen for chemicals and small molecules to see what converges to rescue all of these phenotypes. And interestingly, they found that estrogen compounds rescued the phenotype, and that this was also conserved in human brain in vitro models. So this is a good example of how Xenopus offers the potential to uncover the conserved biology between human brain disorders, as well as move towards rational drug design. And in terms of how ZenBase supports that, is we're curating all those phenotypes with ontologies and then linking them to human diseases as well.
Okay, cool. I mean, I think maybe this happens later in the meeting, but what ontologies do you use in ZenBase?
So we curate our phenotypes with our Xenopus phenotype ontology, which is almost completely eupheno compliant. We link to human disease with the human disease ontology. We have our Xenopus anatomy ontology with cross-references to Uberon. So
cool. And then to finish off with Yvonne, zebrafish.
Well, zebrafish has been used for the gamut of human disease modeling. As someone had asked earlier about high throughput screening using the Cas9 system, and there is definitely a lot of work going on with that. I haven't seen a particular example more where they're screening large genome sets, but Cas9 has been used for Saul-Wilson syndrome. They've knocked out COG4 to look at the developmental contributions of COG4 to see how that is contributing to the cellular pathogenesis of the skeletal dysplasia. Additionally, zebrafish are used significantly in cancer research through xenograft and transgenic overexpression of human genes, and as well as in drug screening, drug discovery. A lot of high throughput applications to investigate drug discovery in zebrafish have become very important in that area.
Yeah, I mean, I guess the fact you can just put it in the water, things
right,
like that really helps.
Yes.
Yeah. Okay, and then to finish off with the final question was like looking to the future, how can we make the data more useful to the research or medical community for each of these resources? So yeah, starting with Pilar again for IMPC.
Okay, so I think one of the things that could be more useful, and since the IMPC relies in several of the Monarch algorithms and tools, like obviously the HPO and Phenodyne, to try to perform comparisons between the phenotypes observed in the mouse and the phenotypes observed in patients. I think one tool that could be useful and could be integrated within the IMPC resource will be like being able to use as an input a set of HPO terms, a set of phenotypes observed in the patients, and just get the mouse models that might be more similar to this set of HPO phenotypes that we are providing. So that I think that could be a way to identify very quickly potential mouse models, potential genes associated to that set of phenotypes. And another thing that I think I could be useful, and we are somehow already working towards that, is to being able to correlate phenotypes observed in the mouse embryo with prenatal phenotypes observed in humans. So that will be two ideas that I think could improve the connection between the human genetics community and the resources provided by the IMPC.
Okay, thanks. And then over to Todd. I guess you've told us lots of the future plans coming anyway, but if you've got any extra
Yeah, there's actually just a couple things I want to mention in this context. From our standpoint, we think that probably the biggest need is to make the human and the model data much more interoperable. How can you easily jump from one to the other? And we've got two initiatives going in that direction. One, which is the critical linchpin, is the Center for Cancer Data Harmonization, which is led by Melissa Handel. This is a group that's looking across all the data models, all the vocabularies, all the ontologies in the Cancer Research Data Commons, and coming up with ways to harmonize those, to translate between them, and to come up with recommendations so that in the future, when someone starts a clinical study or a new trial, they can pick ontologies or pick vocabularies that are much more easily integrated in with everything else. The other initiative we're taking on is something called the Cancer Data Aggregator, which is a tool that will use the output of Melissa's group to launch queries from a single point across all of the repositories that make up the Cancer Research Data Commons. So for example, if you were looking for osteosarcoma data, you could just launch a single search and bring back all osteosarcoma data that would be available regardless if it's human or dog or anything else.
That sounds great. And over to Mardi.
Yeah, so what we're working on at Zenbase is really working to enhance our data sharing infrastructure to align with other resources such as the Alliance, as well as looking to the future, maybe some developing tools to enhance the use of Xenopus to study human DNA variants, so support for that in our genome browsers, as well as a way to extract our gene expression data to support gene regulatory networks, I think would be really useful.
Yeah, cool. And then finally, Yvonne.
So for us, we're working on better representation of cancer phenotypes in our phenotype. So ZFIN currently uses we're working with UPheno people to or group to move our phenotype annotations to be more UPheno compliant, and I think there's work there that needs to be done with regard to our cancer phenotype ontology representation, as well as better in the area of cancer research, better documentation for cancer xenographs. So that I think there's a need to better be able to pull out which cancers are, which cancer lines are being used or patient lines are being used to facilitate finding the models, one, but also understanding phenotypic outcomes and drug discovery. So I think there's an area that we're trying to improve.
Okay, great. I'm not sure if I missed any questions in the chat. Nicole or Valerie, did you spot
There's one for you. There's one for you, Damien, but maybe you can look at it. It's from Justin Reese and respond in the chat. But I just want to thank you for running a really great panel. I was like scribbling everything down like a crazy person. This was really helpful for me. Thank you to our panelists.
Yeah, thanks to the panel members. It's always challenging being on the panel, and the virtual one's even harder, I think. So yeah, thanks a lot for putting the homework in beforehand as well.
We did have one question from Melissa. Melissa, do you want to ask your question before we move on to the next speaker?
Yeah, so I guess I just wanted to point out, and this is kind of in response to some of Todd's comments, but I think it's something that many of the data resources are challenged with. And I think one of the big things, and it's a big lift that we have coming up, is really, there's 20 plus years of knowledge engineering in the NCIT, but yet it's not modernized with respect to the types of ontologies like HPO or UPheno that we're using in our algorithms for these more computational needs. And there's also a lot of overlaps and gaps across these different resources. And so I think one of the really big challenges is to kind of do a big refactor and re-engineer these things together, because what ends up happening right now is that in the cancer domain, you're perfectly fine to just use the NCIT, but when you want to combine cancer phenotypes with craniofacial defects, for example, you're really challenged to use them together in a combined way. And so I think the Kids First program is really a wonderful environment to start to make some progress on this collectively, because it's really hampering our diagnostic tools to not have the richness of the NCIT encoded within the more computational ontology resources. So that's part of what we're going to be helping with on the CCDH with the Canine Commons, but I think it really takes a village. So it'd be great to think about how we can work together to improve that landscape.
Great, thank you, Melissa. With that, I think we need to move on to our next speaker. So next we have Deanne Taylor, who's going to first talk to us about integrating cross-species resources in a common fund data ecosystem to inform kids' first genetic discovery. And then Deanne will be leading a panel on cross-phenotyping tools and resources that will consist of members from the Monarch Initiative. So with that, Deanne, do you want to take it away? We see your screen, but we do not hear you.
Sorry, for some reason, mute was not unmuting when I was in screen share. Let me try it again.
Sure.
Okay, now you can see it. All right, so I'm talking, can you hear me okay?
We can hear you, but now we can't see your screen.
Okay, let's try it again. Go back to Zoom, do share screen again. I'm usually not the one with this problem. Not usually. Okay, now can you see my slide now? You can, right?
Everything's perfect. Yes.
All right, great. So, okay, so I'm going to talk a little bit about a project that started off as a very humble project that's starting to blow up a little bit, and which is kind of interesting. I should have actually added my co-PI onto this. I mentioned him later, but it's also Ryan Urbanowitz. I'm going to talk more about the data that the work we're doing now for integrating data, although this is a larger project than just data integration, which I'll explain. So, so as we have found out in what we've seen today, is that modeling single themes of variance in human cohorts is actually difficult. And Wendy, has found finally because she's got the power of, a variant that's popping up against genome-wide significance. But this is something that we've known for a while that was going to happen, because if you've worked in genetics for a while, it's really hard, especially in what we consider complex diseases, to find single causes. There's usually a proportion of them that have de novo variants, but not all of them, obviously not all of them. So there could be different combinations. For example, you could have heterozygous variants that are scattered across pathways or functional association classes or genes that work together that may add up to a pathway of loss of function that's been published before, that kind of thing. And also for cohort analysis, how can we try to explore these complex diseases in a more comprehensive way? And there's a bunch of different ways of doing that, and our idea was to try a different way. But as you know, that using model organisms might be one way to get to development, because obviously the old thing is you can't knock out a gene in a human and watch the person develop in the womb. So we're using model organisms for this, and we've heard already just today about a bunch of different model organism data resources. And there's always the developmental perturbation and regional gene expression. So looking for changes that you've actually made to the organism through examining the phenotypes, and also more subtly, you could go back and look at single cells or even staining at early embryo development, look for gene expression in different key windows, or what they call windows of susceptibility in developmental stages. And if you could do that, you can start, considering when those genes are turned on in certain organs at certain times, is that timing somehow disrupted, or is there something going on with that gene? And we go back and look at the human genotypes and try to see if there's a corresponding change in that gene that might be expressing in model organisms during these windows of susceptibility. So one of the things I'd wanted to do for a while is apply some machine learning approaches to Kids First data. And I have a partner, Dr. Ryan Urbanowitz, who's a fairly new faculty member, used to work with Jason Moore at UPenn. And Ryan is very interested in looking at epistasis, and the way that he defines it is looking at gene-to-gene interactions, 2 or more genes that are contributing to a phenotype. And he's come up with several different. His specialty is machine learning, and I've brought him into the light side of looking at developmental physiology and using this to look at birth defects. And he is now an enthusiastic collaborator. So we're working on trying to integrate machine learning from the basic stuff that I can do, which is like, random forests and gradient descents and all sorts of other stuff that's more typically used, to more complex things that Ryan works on, which are using gene sets to look at epistasis. So we weren't just interested in looking at the quote univariate way of doing things with like statistics and looking for single variants, but also using multiple different combinations of variants that might be showing up in the cohort. So, but the problem is when you start doing epistasis types of analyses, there's a computational workload or load on the computation. You can imagine you start adding more and more genes, that the combinatorics can blow up exponentially, because that's the definition of an exponential. So you end up with too much computational burden for looking at these complex collections of variants in the case of, say, collections of heterozygotes that are acting together in a pathway to retard or somehow slow down the expression of a gene during a critical susceptibility window, which might then add a knock-on effect further on to generate a developmental defect. So we have this animal model data. We have this idea of trying to use multiple genes and pathways. So how can we leverage model organism data? So, our idea is to essentially try to shrink this giant space down to it's like looking for your keys under the streetlight. It's not always the best way, but it's a way to start. So are there regions of the giant computational space for all possible genes and features in the genome, selecting out different regions based on prior knowledge? And we have prior knowledge already. For example, in MGI, if you search for congenital heart defects for different phenotypes that they have, there are 36 genes associated with this. And this is just some of the genes here. Some of them may be familiar to those of you that work on this. But of course, it's probably more than just 36 genes. But this is one way to get to it. If there was a way to expand out this list of genes to genes that are associated with them, like known protein-protein interactions, et cetera, that might be a good way to go about trying to create a still large, but not completely enormous, overwhelmingly enormous space for the machine learning algorithms to operate within. So there's one way to organize this data. It's called the knowledge graph. This is just a snapshot of one that was developed at CHOP that we did earlier for looking at cancer targets. But this is just a nice, cute little schematic. But this shows you that you can link together information based on the relationship between things. So you have a pathway that's encoded partly by a gene. The gene there's also an agent or a drug that targets that gene. So you can see there's relationships here that are well-defined relationships based on prior knowledge. Experimental data as well. So it doesn't just have to be something that's been named in the literature, but something that actually comes out of a data set as well. So if we can organize our data this way and to make a very rich resource that we could then go backwards to this state where we can actually select out certain areas of this giant space to look for gene to phenotype relationships in humans, that would be awesome. So there actually happened to be a call for applications from NIH based on the Common Fund data sets. So it was in R03. And Ryan and I talked to Ryan, we decided to put in an application, and we were going to focus on the data sets I've highlighted in blue, GMKF, COMP2, which is now part of IMPC. So we're really bringing in all of IMPC, GTEx, links, and HubMap. And the idea was to bring in these data sets together, mostly bring in, not Gabriel and Miller would be the user of the data, people using Gabriel and Miller, but all the other additional data would come in and inform the gene choices we'd like to make. So a use case idea of this is, let me skip forward here. So a use case example would be, so suppose you want to find a set of genes corresponding to a phenotype in Kids First. So let's say heart defects. So first you'd want to find model organism phenotypes that have some sort of similarity to the heart defect you're interested in, or all heart defects, versus the Kids First phenotype of heart defect. And then to determine the model organism's corresponding perturbed genes. and then match the human genes as expressed in GTEx tissues of interest. And I'll explain that in a second here. But why GTEx? Because that's adult tissue, but there is some signal there. You can trim or expand feature sets by phenotypes, variants, gene, or so the feature sets would be genes or variants. You can trim or expand your list of genes and variants by adding all this different data if you'd like. Let's say, let's just add more phenotype data. Maybe we want to add another phenotype in there, or we want to add variants or gene pathway annotations or gene-gene interactions. And so we could, we could add additional genes to enhance and enrich that pathway. And then we could trim or expand that data set by gene expression. So let's say you want to say, oh, I only want to see things that are expressed in pediatric heart. So then you can exclude out the genes that are not in pediatric heart. And then you could take those features in terms of genes or variants and say, okay, I'm going to go back to the Kids First data with my shopping bag full of genes. I'm going to apply my machine learning back onto Kids First data and see what I come up with. And it turns out that, things like GTEx, we've applied this method that I've just talked about using Go, starting off with Go categories as our gene sets. And when we applied this to the Pediatric Cardiac Genomic Consortium data, PCGC, we found that when we use univariate types of analyses, like the typical GC, we did find one pathway that seemed to be related to cardiac function. When we applied this across the genotyped cohort, so these were chips and not exomes or whole genomes, but they were genotypes. We applied this to our analysis. We found many more cardiac categories coming up natively. This was not at all affected. This is all from the idea of the multiple hit hypothesis in the machine learning. So we were finding some very interesting data on Go. And that Go is not GTEx, but Go is also not necessarily predicated on development, and it's also often based on model organisms. So there's possibly other functions that could come out of GTEx, pardon me, that are showing up here in these pathways. So, we feel confident that bringing in GTEx would maybe help, especially if there are things like master regulators that affect overall transcription in the organ. How many more minutes do I have left, Nicole? My clock disappeared. Another 2 minutes or so, or am I done?
We're hoping to end the session at 11:15. So yeah, maybe if you could wrap up, that would be great.
Okay, so essentially, we were using the UMLS Unified Medical Knowledge System, which integrates a lot of different ontologies and vocabularies. It has its problems as an ontology, but it also is a unified system that we could get to really quickly. We only have a year on this RO3. We're working with Jonathan Silverstein and Chuck Borromeo at Pitt and HubMap, and they had already come up with a knowledge graph on the Unified Medical Language System from NIH, which includes phenotypes and a lot of different annotations. And we're starting to integrate data already onto the existing UMLS graph that Jonathan's created. He's also using it for HubMap. But the difference is that we're starting to add in model organism data onto the human data through connections that are actually coming up through work that we're doing, partly with people from Monarch and partly with people like Justin Reese or at least advisement from people like Justin. I'll skip through here just saying that we are able to, it is now a functional database. We've already integrated the model organism data and are in the process of refining that. And we're also adding in GTEx data this month. And these are our collaborators. So collaborators and helpers. And I probably left some out because this is becoming a much more expanded project than I thought it would be, because it is successfully working. And I think a lot of people have interest. So that we'll have a product where you can actually integrate data on the fly and name what your outcome should be. So with that, I'll just put up the panel slide, and if anyone has any questions, you can feel free to ask.
There's one question in the chat from Heidi Sophia about polygenic risk scores being used as part of the CF Knowledge Graph project.
Yes, we can do that. In fact, interestingly enough, because polygenic risk scores are not, I mean, you could try to generate them, I guess, but that's not what, obviously that's probably not what you're asking, but we at this moment don't have polygenic risk scores as part of the Common Fund, and this was supposed to be around the Common Fund data, but I snuck in one of my slides that we're going to be integrating links and other data sources. So there are polygenic risk scores that we will be adding in as part of a variant collection from different literature sources, at least that I know of. But if anyone has any recommendations for good papers for that, I'll take it. I only know the ones right now that are related to cardiac defects and cardiac malformations on the maternal and fetal side. But I mean, we're going to look for others. Anything else? Okay, so should we start in the panel?
Yeah, that would be great. If we could hold the panel session until about 1/2 that would be great.
Okay. So I don't have an introductory slide. I don't know if you had one, Nicole, originally up there, because this is not necessarily your slide.
Yeah, no, this is perfect.
This is okay?
Yep.
All right. So Justin, do you want to introduce yourself, and Kent and Deepak? Or
Sure. I'm Justin Reese. I'm a computational biologist at Berkeley Lab. Deepak Uni's on the call, and Kent also. We're all members of the Monarch Initiative. So I guess we're going to focus on telling you more about the resources that Monarch can provide for solving these kind of problems. And I guess maybe we could give first a kind of an overview of what data exactly we integrate. So
you want to share some slides?
Sorry, I don't have any slides. I could.
It's okay. Okay, that's fine. Yeah.
Yeah. So Monarch, I think that probably the from where I'm sitting, the main resource that Monarch brings to the table is the knowledge graph, or at least me. I'm sort of more of a programming type guy. It integrates data from at least 19 different species. All the big mods are represented here, and we ingest from them on an updated monthly basis. ZFIN, Flybase, NGI, IMPC, Wormbase, just probably too many to mention, and a lot of ontology resources that you heard about today, HPO, Go, and then a lot of human resources that are useful in this context too. Obviously, ClinVar, OMIN, GWAS, Catalog, Orphanet, and it's probably too many different kinds of data to enumerate all of them, but phenotype, genotype, disease, PPI, gene function, pathways, it's really just a trove of information for doing the sorts of things that you heard about today. So the knowledge graph is freely available for download in RDF format, and also I think in TSV. And then there's also, I guess, maybe Kent can give a summary of what the Monarch UI provides. So the Monarch UI obviously is pulling data from the Monarch knowledge graph, but that also is a really rich set of tools that provide tooling for the sorts of things you heard about today. Kent, what am I missing?
That sounds great. Yeah, I could give a quick demo of Monarch if that'd be useful.
Yeah, I can unshare my screen.
Okay. So I'll try not to spend too much time, but this is our website. And yeah, I would say the biggest parts about our site is data exploration and our phenotype analysis tool. So I'll start the phenotype analysis tool, which is probably of interest of this group. So basically what we're doing here is taking a list of phenotypes and comparing it either to diseases or model genes. So I'll go through a very contrived example. If you have HPO terms, you can enter them. We can also search for other genes or diseases to just get a starting point for phenotypes. So here I'm just gonna look for diseases and look at HPO terms associated with NF1. And it should auto-populate. So yeah, we have a large list of HB terms here associated with NF1. You can adjust or remove. I'm just going to confirm it. And then for comparisons, what I'm going to do is look for similar mouse genes. I'm going to click here a little fast. And what we get is a heat map of results. I'm going to swap the axes real quick because it just looks a little. Yeah, so on the x-axis, we'll see our HBO phenotypes that we had input, and then on the y-axis, the mouse genes that it's matching. And this is one example where we'll actually see NF1, the mouse NF1 show up at the top, or actually tied with NF2. And in the heat map, you can click these and see the phenotypes we input for NF1, so spina bifida, and then where it matches in the mouse phenotype ontology, or I'm sorry, mammalian phenotype ontology, which is exencephaly, and then the term in common, which is neural tube closure process quality. And we're working to update UPheno, Shanika will talk a little bit more about, to improve these matches. So this is one way to explore our data if you have a list of phenotypes and want to explore similar diseases or model genes. The other way is to just enter things into our, it's a little smaller, our search bar. So I think earlier there was a discussion about heart defects, phenotypes. So I'll put that in and hope for the best. So this finds abnormal heart morphology, which I assume is a synonym. And over here, we'll get a bunch of genes, variants, diseases that are annotated to abnormal heart morphology. And we're working to improve these views. So this is what we consider a parent term in the HP ontology. So we'll get a lot of subclasses and genes kind of repeated, linking to subtypes of heart defects. And so, we are working on, better showing this data and filtering. We'll see some human genes here. I think I know if I go to the very back, we'll see mouse genes, but we're working on a widget that'll let people just select these and focus on these different genes or different species. So this is a general, this is another way we use the ontology. We we'll look at the subclass relationships to show data and their associations. That's the gist of it. We also have a NLP tool that can also lead into that phenotype widget I was showing earlier. And so another way to explore data is by putting free text into here. And I don't take too much time. I'm going to stop sharing and pass it back.
Okay, and Deepak, do you want to introduce yourself, or should I go into the questions? Or
yeah, so I'm like Justin, I'm also a software developer here at Lawrence Berkeley Labs, and I've been working with Monarch for the last 3 years, and I'm mostly focused on APIs and getting our Monarch knowledge graph used in other projects and consortia, and in that effort, making it more amenable for working with machine learning tools like graph-based machine learning with Justin and Nico, and also Ken. So yeah, we can probably move on to the questions.
Oh. Dan, we should have Nico introduce himself too.
Oh yeah, Nico.
All right. Yeah. Hi. I'm Nico. I'm also working with Monarch initiatives, and I'm mainly their ontology guy for the more technical side of the ontology pipelines. And yeah, we'll talk a little bit about UPheno. So the one thing that makes, of course, an ontology a bit more boring than these exciting tools that we've been seeing all through today is that actually it's not really very user facing. I will still show it to you a bit so that you can get a bit of sense and explain to you also what this whole UPheno project is about. So I'll just quickly share screen. So the first important thing, I guess, is can someone confirm that you see the right screen with some kind of hierarchy on it?
Yep. Yes.
Great, thanks. So the first thing to think is UPheno is essentially two completely different things, I would say. So one, it is an ontology that integrates species-specific phenotype ontology. So in that, it's like an artifact, an ontology like Uberon and like other ontologies. But two, it's also kind of a socio-technological framework that allows the standardization of patterns used in these ontologies to make them more interoperable and able these kind of cross-species computational analyses. And so you can see even if you look at the ontology in a tree form, it's just kind of an abstraction of what actually is all in it, but it's, I think, a useful one. So what you should think of UPheno to be is basically an ontology that is currently focused on phenotypic abnormalities. So you have, if you were to browse it down, you would see up top a bunch of species agnostic phenotypic abnormalities, and then as you browse down further the tree, you would see some phenotypic abnormalities that pop up that actually come from species-specific ontologies. Like here, this is an example from Xenopus, or if you go further down, abnormal growth, you see from the human phenotype ontology, growth abnormality, and you go further down, abnormal cell growth. You can see something from the Drosophila phenotype ontology, from the C. elegans phenotype ontology, or even from the zebrafish phenotype ontology here in a different branch, and the mouse phenotype ontology. So you can see, you could browse further down and see more things. So I'm not going to do that now, but you get the sense of what this is. And the other side of UPheno is the side that is dealing with turning them into a computationally consistent and powerful artifact. We do this a lot through the logical axiomatization, which we will not explain now, but looks something like that. And what we do here is we have this kind of community group that meets regularly, involving many people from across all the mods, some of which have even have spoken today as well. And we sit there and we discuss in a panel patterns for representing these computational phenotypes that can then that are then represented consistently across all these different species ontologies. And yep, lastly, I would say that UPheno is primarily, I would say, used for data aggregation, grouping data from across species, but also for data analysis and prediction more and more. Some of the things that Diane was talking about with the machine learning based approaches, it's becoming more important, and the phenotypic similarity that is used in Exomizer and similar tools are also important for diagnostics.
So some of the panel's questions we have here, I think it's kind of answered. How do researchers use your tool or research to facilitate cross-species research? I guess that's for everyone here. Like, if someone wanted to do cross-species research, for example, identifying variants or identifying genes in their kids' first data set that might have some connection to disease, you know, how would they go about doing that? How would they start? And that can be for any of you. I don't know, Justin or
I would say the most accessible way to do that is through the Monarch UI, which Kent just showed you, which is really just a beautiful way of exploring the Monarch KG.
Yeah, Nico.
So I would I would add that there are many different ways, like it depends a little bit on how much resources you have. Like, one of the most powerful things I think that is totally underexploited, and if people were to start doing it, I think that they would reap a lot of fruits here, is to actually use the knowledge graph to start doing more advanced machine learning techniques, like the things that you've been proposing before in your talk. So there's a definite gap here, that we have done a lot of very careful engineering work on the knowledge graphs on the ontologies, but not a lot of exploiting that work for computational analysis. So I would say, I would recommend, if you were interested in cross-species analyses, to actually look into using and reusing our knowledge graph and ontologies directly.
I'd add to that, we're developing tooling, Nico and Deepak and others are developing tooling to do just that, so to make graph machine learning more accessible. We have this tool that we're developing called NEAT, which is a way to apply machine learning on a knowledge graph using using YAML. So you just basically describe what you want to do in YAML format, so you don't have to do any programming. I mean, it doesn't, of course, remove all the black arts involved in machine learning, but you can at least describe it without doing programming.
And also, like, just to access the knowledge graph, I think the UI is one place, the other is the BioLink API or the Monarch API, which lets you do more programmatic queries to do some bulk analyses that otherwise would be difficult from the UI. Yeah. And ask a very interesting question and query different data types according to how, according to the answers that you get and how you want to explore.
So if they were going to start with, I mean, they could start also going to the Monarch resource itself, correct? I mean, because there's a lot of good resources on the website as well. I know that's not directly what you guys are involved in, but you know, we could also encourage people to go to Monarch and start exploring at least the data that's there as well. So has any work that you've done informed a clinical diagnosis? So are you involved with any kind of clinical groups, or even if it hasn't informed one, are you busy working with clinical groups that link, you know, human data back to model organism data?
I think Damien described probably the best example of that in glorious detail about the wonderful Exomizer tool, which leverages part of the Monarch KG to do its thing. There are also reports in literature of other people sort of in the wild using the Monarch KG to you know, apply this knowledge in the clinic. Yeah. Lyrical also arguably uses the Monarch KG. This is a tool that Peter Robinson wrote, which is not entirely dissimilar to Exomizer.
So I would add that one key feature of the Monarch knowledge graph and its various ontologies is that that everything that we do becomes amenable to semantic similarity analysis. So, for example, in Exomizer, in order to actually go from mouse phenotypes to human phenotypes, it's as we know, these communities are very, very different. They use very different terminology. So it might be that that, for example, one terminology would one community would call a phenotype brachydactyly, another one would call it short digit. You can't really, there's no way to automatically match these together, but because of these kind of axiomatization and the graphs that we are building, we can bridge those using semantic similarity algorithms and in various ways. And I think that this is something that's really important for tools like Exomizer, and also for merging actually these separate, let's say, let's call them data sources like MGI and MPC on the one side and the human stuff together. So I really have a lot of hope for this whole ontology mapping stuff that comes out of also some semantic similarity informed approaches.
So another question, and I think this is kind of something that's been addressed a few times, but where are your tools going to be going towards making it even more useful to the research community or the medical community? And I think that comes back to what's already been presented, but I don't know if you have any additional ideas.
I think the one I'm most excited about is what we just discussed, which is applying and developing better graph machine learning algorithms to the knowledge graph, because the data is all really there, and there's a lot of latent knowledge in there that is very be an awesome substrate for a lot of these algorithms. There's a couple of challenges there. Not least is the sheer size of the knowledge graph, and so that's sort of an active area of research. Is it's easy enough to extract, you know, to do feature extraction and extract a subset of that. Well, maybe it's not easy enough, but it's very doable. But it'd be better still to just throw the whole knowledge graph at a graph machine learning algorithm, but it's a bit too big depending on what algorithm you're talking about now. And so we're excited to develop some algorithms that can deal with knowledge graphs on that scale.
So I'm super excited for the process of trying to see whether we can mitigate some of the let's say variation or variability in curation practices using patterns. So we see this all the time, like some sources, let's say they use an approach where they have an implicit abnormal concept in their annotations, and they use anatomy terms to annotate their data. Then other groups use terms from phenotypic from ontologies about phenotypic abnormalities. Again, other groups, for example, from the GWAS side, they're using traits and qualities for annotating their data. And I think that these kind of pattern-based approaches that we are kind of spearheading right now could be used to bridge some of this stuff together. So concretely, for example, you could think of this problem of bridging from anatomy-based curation pipeline to a phenotype curation pipeline as a scenario where you actually just cast the phenotype, the anatomy data coming from this source into a pattern of abnormal anatomical entity phenotypes, and that way you can actually make those two things comparable directly in a knowledge graph. So this kind of thing, and this we are actually working actively to do this for traits as well, which is much more difficult. And yeah, hopefully this year we will see some results of doing something like that.
So what do you think about how are the underlying ontology structures? I guess this is more of a technical question. How does the underlying ontology help with query, display, and retrieval of the data is the direct question. I think this is, I'm not sure if the underlying ontology means the underlying ontology structure or the actual ontology itself. But how do you see your ontology work that you're doing actually fit into the knowledge graph, or maybe fit into the overall picture that Nico was just discussing? How does that help with, how would you say it actually helps people with, I guess, query, display, and retrieval of data?
Yeah,
I know that's a field-wide question, right?
Yeah, it is wide. I mean, there are many different things. So I would say in the most trivial answer is our ontologies provide species independent or at least species agnostic terms that subsume species specific ones. So if you wanted, for example, to find everything, all annotations, everything you want to know about some specific kind of abnormality, let's say abnormalities of the circulatory system, and you wanted to know what do we know across all the model organism communities, you could use that to expand, to aggregate and group your data. That's one thing. The logical structure can be exploited in many ways. So one we've discussed before, semantic similarity, but other things is you can actually query this information with post-compose queries that are not necessarily corresponding to phenotypes. So you could ask, for example, give me all the, let's say, circulatory system phenotypes that have something to do with abnormal cell proliferation or something like that. This doesn't have to be an actually existing concept. So you can just phrase this as a post-compose expression, and then you get all the phenotypes across all the species back with regards to this query. So these are two ways, for example.
Ian, can I ask a question? This is Valerie.
Yeah, good.
Kind of to follow up to Melissa's two comments in the chat. You guys are very technical heavy, but you're creating this resource, these resources for the whole community. But would you be open to, direct collaborations with research groups who've never used ontologies before or never used these tools before, to sort of help them address the, especially some of the use cases we heard about this morning?
You just broke off, but I assume you stopped talking. We love generally to do that. We do this all the time. So model organism communities of all kinds. So just now we've been helping some of the insect communities to come to build their anatomy ontologies in a way that is more compatible with Uberon, for example. And we do this generally all the time. And yes, we are very, very open. It's been, in fact, we seek this. So if groups are interested in building their own vocabularies and they want to get some advice how to build those in a way that makes them inherently from the onset compatible with all the other vocabularies that are already out there, I would definitely recommend to contact Melissa or contact us directly, and we will help you get started with this kind of stuff. Yes.
Okay, looks like I think we're about almost at half past the hour. Is there any other things that you guys want to add towards the end to sum up, or?
I think that I just want to re-emphasize that there is a lot for the picking there. There is a lot of stuff a lot of great engineering work that has been going on that is not yet exploited. And they think that some of these more predictive analysis, getting candidate genes, there's still some work to do. For example, one thing that I wrote down here before is just like, is there any way we can quantify and sort of determine what makes a great model? This is like a really difficult one to answer. So obviously if you analyze cross species in a relatively simplistic framework like Eupheno, you have the problem that you integrate species that are taxonomically quite distant from each other, and there's obviously some kind of noise that will be introduced by that. And here there is some work to be done, but there's a lot that can be exploited already. And I hope that people will pick up these tools and start running their own analysis and tell us also, look, guys, this doesn't work. This is really bad, and we will fix it. So that's one of the things I would say.
And
important.
And I know that Melissa in the past, and she also says this in chat, is that people in Monarch would be really happy to work with other investigators to help annotate and integrate their data to be more reusable. And I've heard her say that in person many times. So I'll go and support her on that one. And the entire Monarch team, I know it's been really a delight to work with on my side, and I'm sure other people will find you in the same way.